Thank you for this opportunity to talk to you today about an ongoing research project in my lab in which we're examining DNA methylation in cells from milk. But first let me say methylation is simply the addition of a methyl group, a carbon with 3 hydrogens, onto the DNA. In a normal cell, in the promoter region of the DNA of a gene, there's very little methylation in the promoter region. That means that when the DNA receives the appropriate signal, it can be turned on and RNA and a protein can be made. But in a cancer cell, there's the addition of these methyl groups in the promoter region, and that frequently stops the DNA from being turned on, and so therefore no protein is made. When that protein is important in cell cycle control or repairing DNA, the cell can then take on characteristics of a cancer cell. And so we know that in cancer, many genes, specific genes, we call them often tumor suppressor genes, are methylated. And we also know that this methylation occurs early in disease. And most researchers would agree that examining methylation in breast cancer, in breast tissue, would be a very good way of assessing a woman's individual risk of developing breast cancer. But the problem is, how do you get to the tissue inside the breast? Now, these are some of the methods that have been used in research to obtain cells from healthy women and from women with cancer. And research has shown that cells obtained from fine needle aspirate, ductal lavage, or nipple aspirate give the same methylation patterns when taken from women with cancer as does the tumor cell itself, the tumor cells from the cancer. Furthermore, the methylation patterns studies have shown are associated with risk. So there are methylation patterns seen before women get cancer, but women who do get cancer, and they are associated with risk. However, these methods for taking cells from the breast have several problems. First of all, a few number of cells are obtained. Often the cell number is too low. As you can imagine, it's invasive, and most women would not want to undergo these procedures. And also, they survey just a small area of the breast. In contrast, in breast milk, you have millions of naturally exfoliated cells. They represent all the tissue of the breast, and it's easy to obtain multiple samples, and samples from both breasts, and repeated samples. Women are very generous with the breast milk. The problem to date has been, though, we haven't been sure whether or not the cells that we obtain from breast milk and the methylation signals that we see in that are indicative of any risk, or do they simply tell us that we came from a lactating breast? And so the results that I have in the next slide, I think, show that the cells that we obtain from breast milk tell us about risk. So let me first, before I tell you about the study that I'll be presenting here today, tell you a little bit about two populations. In an early study, we collected milk from 102 women at no increased risk of developing breast cancer. We examined the methylation pattern. We separate out the epithelial cells from this milk, and we examine the methylation pattern. And that's what you see over here on the unselected population here, these blue, the blue bars. And what we found overall, that these women, these healthy women, had very low levels of methylation in the promoter region. We examined 12 genes, and that's what you would expect. They were mostly healthy women. But the dots there represent outlier values, and we did see a number of women that had slightly higher values, and these were associated with risk, as based on family history. So let me just, for a moment, since I'm going to show a few more graphs, go over what we call the box and whisker plot. The box and whisker plot, each one of these represents a single methylation site on 3 genes. The top gene is a tumor suppressor gene. The second one is a second SFRP, and GSTP is involved in metabolism of toxicants. Each site on the x-axis represents a specific methylation site in the promoter region of that gene. And in the box and whisker plot, the middle bar is the median. The lower bar is the 25th percentile. The top is the 75th percentile. And those whiskers represent 1.5 times the range. So the dots above that represent individual women then that are outside the range. And so those dots, They can vary from the number of women that are outside that range may vary with methylation analysis with site, and those are true significantly higher methylation scores, and we call those outliers. And we're particularly interested in the outliers. Okay, so for the study that I'm presenting here at AACR today, we wanted a slightly higher risk population. And so what we did, we collected nationwide milk samples from 250 women. They come from 41 states. And the requirement for this study is that the woman be a nursing, be willing to give us milk, but that she also have had a biopsy or be scheduled to have a biopsy. So she's at an increased risk. They send us the milk, they send us the biopsy report, and we categorize them by biopsy report. And we also do long-term follow-up because some percentage of these women will have cancer. And so as you can see right off from the breast biopsy population, the red bars, box plots, and those dots, the medians actually are not different. So those median lines that I told you don't differ between the two populations, which is good. Most of these women are not going to get cancer. But the percentage of the outliers and the height of these outliers does differ for these 3 genes that are methylated in cancer. So now let's look a little bit more closely just at the population on the left, the breast biopsy population. So what we do, and one other thing here, I should mention that if you have the abstract, we've also updated and added in more information. So this, there might be a slight difference in a few numbers. This is the updated. And we update this as the women send us in their biopsy reports. So to date, we've finished the analysis on 3 genes. We plan to do 12 genes. And we have complete biopsy reports from 182 women. Only a small percent have cancers, 13, as you see there. The majority of the women have benign proliferative disease and non-proliferative lesions. And what you see for a single gene I've chosen to show you is RASF1, again a tumor suppressor gene. And on the right side, you can see that the methylation and the outliers for the biopsied breast, and on the left side, you can see for the non-biopsied breast. So we're even getting a specific breast signal. In the panel before this, I showed you that we had 2 different population signals, a high risk and a low risk, and now we have specific breast for the left and right. And if we limit then our analysis to just those women who had cancer and just focusing on a single gene, we see that the biopsied breast is more significantly methylated than the non-biopsied breast. Now, this is a study in progress. We need to examine more genes to develop, to be able to even speak about specificity and sensitivity. I can't talk about that yet. And we need to examine more women, and we specifically need to get breast milk samples from more women who are having biopsies. I think this study has some important implications. I think it's important that young women be able to be screened and know if they're at higher risk, and this is especially important since recommendations now are to delay screening mammography until age 50 unless you're at higher risk. But women don't necessarily, since most cancers are still sporadic, women don't know their individual risk. I think it could help with detection in pregnancy-associated breast cancer, and this is important as women are waiting later into their 40s to have their first child. They're at actually a higher risk of developing pregnancy-associated breast cancer. And finally, methylation analysis and a screen based on methylation analysis is particularly valuable in that methylation is reversible, and I hope that this research will help with an impetus to develop prophylactic drugs that can be used prophylactically, as they are now being used in the clinic for breast cancer, but to use them prophylactically for decreasing methylation. Thank you.